Figure 1 shows an animal cell viewed using a microscope. So here's figure 1, and just below you can see the cell. The question says the cell contains a nucleus. What is the function of the nucleus? So looking back at figure 1, the nucleus is this part down here. It contains the DNA of the cell and all the instructions it needs to be able to carry out reproduction and growth in the future. And so the way you can say this is that it controls the activities of the cell and it holds its genetic material. So either one of those statements, controlling it or holding the genetic material, will get you that mark. Name one type of cell that does not contain a nucleus. So there are a few answers you can potentially give here. One is the red blood cell, and this doesn't contain a nucleus because it needs that room for hemoglobin. Alternatively, you could mention bacteria or any other kind of prokaryote, and these don't have a membrane-bound nucleus. Instead, they have their chromosomal DNA free in the cytoplasm, and they can also have loops called plasmids, which contain genetic material. And then you can also mention the xylem cells, so those would be in the plants. And they don't have a nucleus because they're not living cells, they're dead cells just acting as a tube to carry water, and so they're not carrying out any functions that a nucleus might be needed for. Draw a simple diagram of the cell in figure 1. Label 2 parts of the cell. So if we have a look back at our figure 1, we've already got some key components. We've got that general outline over there, which is going to be the cell wall, and we've also got the nucleus, which we mentioned earlier. So what the mark scheme is looking for you to do is draw the approximate same shape as the cell we see before. You can label the outline as the cell membrane, so that controls what goes in and out of the cell. We can also draw the nucleus, so that was quite a clear component in the diagram, and that controls the activities of the cell. So both of those will get you a mark, and you only need 2 components. Alternatively, you can label the cytoplasm, so that's where your chemical reactions might be occurring in the background. You can label a mitochondrion, the site of respiration in the cell, and you can also draw a ribosome, so this would be smaller than the mitochondrion, and it's the site of protein synthesis. Name one structure found in a plant cell but not found in an animal cell. So there are a few potential answers here. You could mention the cellulose cell wall, which isn't needed so much by the animal cells because animals have skeletons, whereas the plants need that cell wall for support and keeping it rigid. Alternatively, you could mention chloroplasts, so these aren't needed by animals because animals don't photosynthesize, plants do. And then you might also mention the vacuole or the permanent vacuole, and these are needed more by plants than by animals because they contain water and cell sap and they help the plant maintain rigidity, whereas an animal cell doesn't need to be so rigid or under such pressure. Figure 2 shows some different cells. So here's figure 2 below, and we've got a group of cells and we have this label X through Y. The question says the real length from point X to point Y is 0.06 millimeters. Calculate the magnification. And it gives us this formula we need down here. So magnification equals size of image over real size of object. And the question is worth 3 marks. So what you want to do here is get your ruler and measure this length from X through to Y and then just record that value, because this is how you're going to get your image size. And it would have been printed to a specific size on the paper. So in this instance, the size you would have measured is 24 millimeters. And the reason we do it in millimeters as opposed to recording it in centimeters is because the question gives it to us in millimeters, and so this saves us having to do that conversion. So back to the question, we've now recorded our size of image, so that was our 24 millimeters, and then the real size is given to us by the question, that's the 0.06 millimeters, and then we just plug that into our equation. So 24 over 0.06 will give you the magnification. That will tell you how many times bigger the microscope has made the image. So 24 over 0.06 gives you an answer of 400 times. And your marks would be for making that measurement of the 24 millimeters, putting your equation together correctly, and then getting that final answer. The cells shown in Figure 2 were viewed using a light microscope. Give 2 advantages of using an electron microscope instead of a light microscope. So electron microscopes work by firing a beam of accelerated electrons at the specimen, and then these get reflected back and detected by the microscope to create an image. The electrons actually have a shorter wavelength than light does, and this means that they can get into those spaces and detect the smaller features, which means that the electron microscope therefore has a higher resolving power, so it's better at telling the difference between 2 small objects and identifying that they are 2 things rather than just one, and it also has a higher magnification power. So those last 2 are what you can put for your answer. You can say that it has a higher magnification, and then you could say that it has a higher resolution or a higher resolving power. Another thing you can potentially say is that it gives you a 3D image, and this is because with the electron microscope, as opposed to just looking through the specimen as you are with a light microscope, this one's actually detecting the electrons that are reflected off the surface of the specimen, and that gives it a perception of depth. Mosquitoes carry a pathogen that causes malaria. What type of pathogen causes malaria? Tick 1 box. So the answer to this one is a protist, and specifically it is called the Plasmodium protist. Mosquito nets can help prevent the spread of malaria. Table 1 shows the results of a study in one area of Africa. So here's Table 1, the left-hand column says the total number of people in the study, so that's everyone involved. The next one is the number of people who use mosquito nets when sleeping. And then the next one along is the percentage of people with malaria, and it's divided into those who use mosquito nets when sleeping and those who do not use mosquito nets when sleeping. A newspaper made the following statement: "Study shows mosquito nets are scientifically proven to prevent malaria. Give 1 piece of evidence that supports the statement." So we want to be looking at this section of the table over here, the percentage of people who do have malaria. And we notice that the percentage of people with malaria who do use mosquito nets is only 1.2. Those who do not is 40%. So this shows us 1.2 is a much smaller percentage than 40%. So those who use nets make up a smaller percentage of those with malaria. And that's the kind of statement that the mark scheme is looking for you to make, that there is a lower percentage of people with malaria when they're using nets. Suggest 1 reason why the statement may not be valid. So there are a few answers you can potentially give here. One is that when you look at that table, some people who use mosquito nets do still have malaria. It's not 100% effective. Another answer you could give is that the data is only for one area of Africa, and you don't necessarily know if the trend is the same everywhere. Another thing you could say is that the group size is too small, and you perhaps don't have enough data to draw that conclusion. And lastly, you can argue that no other information about the people in the investigation is considered, so there could be other factors regarding whether you get malaria or not. Table 2 shows information about the number of deaths from malaria in the same area of Africa. So here we have Table 2. The left-hand column is the year, and it goes from 2005 through to 2015. Then the right-hand column is the number of deaths from malaria per 100,000 people. Predict the number of people per 100,000 who died from malaria in 2017 if the trend stayed the same. So here we want to start by having a look at the trend over the last few years to see if we can predict what's going to happen in the next 2 years to get to 2017. So from 2011 to 2013, the number of deaths per 100,000 decreases by 3. Similarly, from 2013 through to 2015, it goes down by 2. So the next jump we want to make wants to be decreasing by 2 or 3. So as an example, we might decide to make the prediction that the number of deaths from malaria per 100,000 people will decrease by another 2 in the next 2 years. So we will take our current value. In 2015, 92 people died from malaria per 100,000, so that's our starting value. We take away 2, as is our prediction for the next couple of years, and that gives us a value of 90. So we might say that in 2017, 90 people per 100,000 will die from malaria. Now remember, this is a prediction that we're making. The trend we've seen so far has been a decrease of 3 and then a decrease of 2. So you as the candidate might decide to predict that it's going to stay around -3 or -2, or you might follow that decrease and go for -1. And because of this, the mark scheme has been written to allow any value between 88 and 91, because we are making a prediction and there isn't a certain number. But the value we calculated in our example, the value of 90, is perfectly within those parameters, so you'd get a mark for that. Use of mosquito nets has helped to reduce the number of deaths from malaria each year. Suggest one other reason for the reduced number of deaths from malaria each year. So there are a couple of answers you can put here. One is improved healthcare. Another is the use of mosquito control methods. And then there can also be different behaviours among people, so changing the way that people act so that they get bitten less often. Describe how the human body prevents pathogens from entering and defends itself against pathogens once those are inside the body, and it's worth 6 marks. So when going for full marks on a question like this, you've got to make sure that you're making your points in a logical order, and then you've got to explain them so that the examiner really knows you understand what you're talking about. And then you've also got to make sure you include both of the bullet points given to you in the question. So let's start by considering the first bullet point, how the body prevents pathogens from entering into it. The first thing that you could talk about is the skin. So this acts as a physical barrier, and your keyword here is barrier, so make sure you mention that specifically. Next, you might go on to talk about this oil on the surface of the skin, and this has the property of repelling bacteria and pathogens. It's also worth noting that this oil can be called in the mark scheme sebum, so if you see that, it just refers to the oil on the surface of the skin. Now you can start to talk about some other forms of protection. So when the body gets a cut or a graze, a scab will form over the top of it, and this also forms a barrier to stop the pathogens coming in direct contact with the bloodstream. You can also mention that the eyes produce tears, and these tears are antiseptic, so they will kill any pathogens trying to enter. And you can get a mark for either of those, saying that the eyes produce tears and then mentioning that they're antiseptic. And then the eye also contains an enzyme which will kill bacteria, so that's another feature that you could mention. Then there are marks available for you talking about your breathing system, so all to do with your nose and your trachea and your bronchi. And what you can say about these is that they contain mucus. Mucus is a sticky substance which will trap the pathogen and stop it from going all the way into your body. The mucus is then carried away by the cilia, so that can be carried back up so that you can spit it out, or it will go down into the stomach to be dealt with by the hydrochloric acid. And the term cilia is just referring to all of these small hairs that line your air passages, so your nose and your trachea and so on. So overall you can see there are multiple marks available for the prevention of entering, but just remember that just listing 6 marks or 6 bullet points isn't necessarily going to get you all 6 marks, so ensure they are logically ordered and explained well, and you're including both of the bullet points. So we can move on to the second one now, defending against pathogens once they're inside the body. So when a person is infected with a pathogen, one of the first things that's going to happen is the immune response, and this is caused by your white blood cells and your immune system, and so that's the first mark that you can go for the second part of the question. Then you might go on to talk about the production of antitoxins, so this is done by the white blood cells, and the antitoxins will neutralise the toxins that are released by the pathogens, so it's battling the parts that actually make you feel ill. Similarly, you can talk about the production of antibodies, and these are responsible for destroying the pathogen itself, so those statements will get you another couple of marks. Then lastly, you can talk about what's called memory cells, and these are a part of your immune response, So they recognise a pathogen that you see beforehand, and then should that pathogen re-enter the body, it will trigger a more rapid response to it, and it will help you to recover from the infection faster than you would have initially. This question is about photosynthesis. Complete the word equation for photosynthesis. So on the side of the reactants, you've got carbon dioxide and water, and then on your products, you've got glucose. A student investigated photosynthesis using pondweed. Figure 3 shows the apparatus that the student used. So here you've got figure 3, you've got some pondweed and it's situated in a beaker of water under a funnel, and the funnel is guiding up bubbles into a measuring cylinder. So those bits that are highlighted in yellow are the bubbles, and the measuring cylinder catches these and effectively measures the gas that is being produced by the pondweed. This is the method used. Set up the apparatus as shown in figure 3. Switch on the lamp. After 20 minutes, record the volume of oxygen collected in the measuring cylinder. Then repeat steps 1 through 3 using bulbs of a different power output. What was the independent variable in the investigation? So first let's just be reminded of this term, independent variable. So this is the variable in the investigation that you are changing. And the method's told us what this is because the last step said repeat the steps using bulbs of different power outputs. So that's what's going to be different each time. So we tick that top box. Suggest 2 ways the method could be improved so that results would be more valid. So there are several options that you could go for this one. One might be doing repeats and then calculating a mean value. Another might be controlling the temperature of the water that your pondweed is in. And you only need 2 points to get those 2 marks, but there are some alternatives as well. So you can make sure that you're controlling the distance between your pondweed and your lamp, and this can have an impact because of inverse square law. Then you can be controlling all of your factors to do with the pondweed itself. So for example, how old it is, how long it is, what the species is, what the mass is, just making sure it's the same that you're using for each part of the experiment. And then lastly, you can be giving it time to equilibrate, which means just getting it used to that new light level that it's at. Table 3 shows the students' results. So here you've got table 3. The left-hand column is the power output of the bulb in watts, and then you've got your central column of the volume of oxygen collected in 20 minutes. Then the last column is the rate of photosynthesis in centimetres cubed per hour. And the question wants you to calculate the value of x in table 3. So here is x in the n column. We're calculating the rate of photosynthesis in centimetres cubed per hour for a bulb that has a power output of 150 watts. So looking at this, we've got our known value of 1.1, and that's the volume of oxygen collected in 20 minutes. So that's 20 minutes worth of photosynthesis, and we're looking for x, which is an hour's worth of photosynthesis. So how many sets of 20 in that 60? Well, that's 60 over 20, which is 3. So 1.1 multiplied by 3 is 3.3. That's how many centimetres cubed are produced in an hour. So that's the answer you're going to get there. Complete figure 4. You should label the x-axis, use a suitable scale, plot the data from table 3 and your answer to the previous question, and then draw a line of best fit. And the question is worth 4 marks in total. So here we've got figure 4, it's an empty graph and we have to complete it using the table on the left hand side. And in order to do that, you've got to fill in your x-axis. So as a rule, the independent variable will always go along the x-axis, and the dependent variable, so that's what you're measuring, will go onto the y-axis. In this instance, the independent variable is the power output of the bulb in watts. That's what we're changing as we go through our experiment. So we want to fill that in at even intervals along the bottom of the graph. So our smallest value is 60, and then we've got 100, 150, 200 and 250. So most of them go up in 50s, and if we test that out on the scale of our graph, we can see that that actually works out quite nicely for us. Just don't forget to specifically add that label of power output in watts. Okay, so now you can actually start to plot your points. So the first one will have an x value of 60, and then it will have a y value of 1.5 because it's the rate of photosynthesis in centimetres per hour that we're going up in. Make sure not to accidentally start plotting the values for volume in 20 minutes. So we work our way up from the value of 60 on the x-axis, and then on the y-axis we plot along at our value of 1.5, and then we make an x where they cross. So you just repeat that process for each of the points. So the next one will be 100 and 2.4, then your 150 and 3.3, 200 and 3.6, and then 250 and 3.6. Once you've got all of them down, you then do a nice smooth curve of best fit, and it should go through all of your points. Determine the expected rate of photosynthesis with a bulb of power output 75 watts. Use figure 4. So what you've got to do here is find the value of 75 watts along our own scale. So 75 watts will be right between the 50 and the 100 on our own scale. And once we've got that, we work our way upwards until we intercept our line of best fit, and then where that crosses, we then find the corresponding y value, and that will give us our answer. So in this instance, we're getting 1.9, or just a tiny bit below it. So we can put that down for our answer. And for this question, the mark scheme will allow you a little bit of leeway, plus minus a small square. So if you got 1.8, that would also be accepted. Which graph shows the effect of temperature on the rate of photosynthesis? Tick one box. So to be able to answer this question, there's one bit of key knowledge that you need to have, and this is that as temperature increases, the rate of photosynthesis will also increase. However, this only happens up until a certain extent, because eventually the temperature becomes high enough that it's actually doing damage to the plant. Things like dehydration and denaturation can occur. So when this is plotted on a graph, you will see the rate of photosynthesis rising upwards and then suddenly decrease at a point. And that is the third graph down, so you tick that box. Water moves from a plant to the atmosphere through leaves. How is the volume of water lost from the leaves controlled? Now, on the underside of the leaf are these little holes that are called stomata, and they open and close under the control of guard cells, which contract and relax. And that's what you need to say in your answer, that the opening and closing of the stomata controls that water loss. Describe the transport of water through a plant from the roots to the atmosphere. And this question is worth 3 marks, so there's 3 points you're going to make. So water is transported in the xylem, and these are hollow dead cells that just go up through the stem of the plant. And then the water will evaporate from the leaves, and the way that it does this is through the stomata, through these little holes on the bottom of the leaf. And really it's these 3 key terms, the transport in the xylem, the evaporation and the mentioning of the stomata, that get you your 3 marks. However, the mark scheme will allow you to also say between the guard cells, because these are what border the stomata. A student investigated the volume of water lost from 2 plants of different species. Both plants were kept together, so in the same place, and figure 5 shows the student's results. So here you have figure 5. The lower down line is plant B, and then the one that's a little bit steeper is plant A. And then on the y-axis you can see the volume of water lost in centimetres cubed, and then the x-axis is time in hours. So the main thing we really see from this graph is that plant A is losing a greater volume of water than plant B. Suggest one reason for the difference in the rate of water loss from the two plants in the first 2.5 hours. So there's a couple of answers you can give here. One you could say is that plant A has more stomata, so these are just holes through which water loss can occur. You could also say that plant A has more leaves, and what this does is increase the surface area of that plant, and it gives it more area from which water can leave, and there's more stomata on the bottom of those more leaves. And then similarly, you can say that plant A has larger leaves, and this has the same sort of effect as more leaves. It increases the surface area of that plant. Both plants were moved to a different place at 2.5 hours. Calculate the rate of water loss per hour in plant B from 2.5 hours to 3 hours, and give your answer to two significant figures. This question is worth 3 marks. So looking back at our figure 5, we want the rate of water loss per hour for plant B after that 2.5 hours, because the graph only actually goes up to 3 hours. And so what we're looking at is that last segment where the gradient gets steeper on plant B, and you can quite clearly see 2.5 hours. It's where the gradient suddenly does that quite sharp change. So what we've got to do is find the amount of water lost in that time period, and the volume of water lost is what's on the y-axis, so that's the difference we want to do. And so we're going to see how much each of these boxes is worth. So 5 of the little boxes is 2 centimetres cubed. So in that space we've got 5 boxes, another 5, and then we've got 3 boxes. So that's 2 centimetres cubed, 2 centimetres cubed, and then 1.2 centimetres cubed. So if we add those together, we get the total volume of water lost. And that is 5.2 centimetres cubed. So that's how much water was lost between 2.5 hours and 3 hours. So back to the question, we've got our amount of water loss between 2.5 and 3 hours. That's the 5.2 we just calculated, and that gets you your first mark in this question. That's the first step. Next, we've got to use this to calculate the rate of change. So we take our change, which is 5.2, and we divide that by the time period, which is the between 2.5 hours and 3 hours, which is half an hour or 0.5. And calculating that, 5.2 divided by 0.5, that gives you 10.4. And that calculation there gets you your next mark in this question. However, 10.4 is the 3 significant figures, and the question's asked us to give our answer to 2 significant figures. So what we've got to do is round the last 4, and that will round down, so your final answer is 10 centimetres cubed per hour. Suggest 2 reasons why the rate of water loss in both plants changed after 2.5 hours. So here you need to be thinking about what affects your rate of transpiration. The environment may have an increased temperature, so it's warmer. It may also have an increased light intensity, so the light there is brighter. And that's already 2 points, enough to get both your marks. Alternatively, you could say that the environment was less humid, so there was less water in the air already. And then you could say it's a windier environment, so the water vapour is carried away more quickly. Figure 6 shows the internal structure of the human heart. One of the heart valves is labelled. So here is figure 6 below, and it's showing us a valve just there. And valves are in the heart to stop the backflow of blood. Sometimes a valve in the heart can start to leak. Explain why a person with a leaking heart valve has difficulty exercising, and this question is worth 4 marks. To answer this question, there are multiple relevant points that you can make. However, you must make sure that you're linking them logically for the examiner. So when you've got a leaking valve in the heart, backflow can occur, and this is when the blood isn't going in the direction it's supposed to be going. So less blood is actually making its way all the way through the heart and getting to the rest of the body. Now when this person is exercising, they need the blood to be delivering oxygen to their muscles so that they can continue. But with this backflow occurring, less oxygen is making it to the muscles, and they can't carry out aerobic respiration to get their energy. Remember, aerobic respiration is the type with oxygen. So now this person's muscles have less energy when they're trying to exercise and the muscle contractions are less efficient. So you don't need to be mentioning every single one of these, we've got multiple relevant points here. However, make sure that your statements are just logically linked all the way through. So you can carry on and you can say that anaerobic respiration occurs. So this is a type of respiration that happens when the body is in a deficit of oxygen. The tissues will start to go into oxygen debt and you get the buildup of lactic acid. And then if we recap this term oxygen debt, it's just the shortage of oxygen in the tissues of the body. You can also potentially talk about the effects of this buildup of the lactic acid. So this is going to start to cause the person doing the exercise some muscle fatigue. Now you wouldn't need to go much further than this to get your full 4 marks, but just another point that you could make is that you then have the less efficient removal of carbon dioxide from your tissues carrying out respiration. So overall, just make sure that you're identifying your relevant points, so everything with a red tick is a relevant point. You explain your points with a logical order, and make sure that there's a satisfactory amount to get the full 4 marks. A patient with a leaking heart valve may have the valve replaced. A study compared 2 different types of replacement heart valve, and these were mechanical valves and biological valves from pigs. The data used in the study was collected from female patients aged 50 to 69, and Table 4 shows this data. So here we've got Table 4, and the column on the right hand side is the type of replacement heart valve, so it's split into 2, mechanical on the left, biological on the right. And then in the left hand column it's got different variables that were measured during the experiment. So the top one is the number of patients given the valve, then we've got the number of patients who died from heart-related problems after valve replacement. We have the percentage of patients alive after 5 years, the percentage of patients needing a second valve replacement within 6 years, and we have the percentage of patients who had a blood clot on the brain after surgery. So the question says give one conclusion about the deaths of patients from heart-related problems after a valve replacement, and include calculations to support your answer. So this question is worth 3 marks. We've got on the left hand side Table 4 to help us out, and the question's asking us about the deaths of patients from heart related problems, which means that it's the second row down that we're concerned with, those values 180 and 178. So because we're comparing between the mechanical and the biological, we've got to be careful with the number of patients given the valve, because it's not the same for both. Mechanical had 2,852 patients, while biological only had 1,754 patients. So what we've got to do is turn our values into percentages so that they're in a format that we can compare. If we start with those who had the mechanical valve, we'll take that 180, so that's the number who died of heart-related problems, and then we divide that by the total, which is 2,852, so that's all the patients who had the valve, and multiply by 100. So this will give us the percentage of patients who died of heart-related problems after receiving the valve. And the answer you're going to get here is 6.31136, and this is approximately 6% if we round that to one significant figure. And now we're going to repeat this calculation for those with the biological valve. So we do 178 divided by 1,754 multiplied by 100. And the answer you get is 10.14823, so approximately 10%. So this question awards you a mark for each of these answers, so one for the 6% and one for the 10%. The final mark is for drawing a conclusion from these numbers, because it says give one conclusion, so you've got to make sure that you make a statement at the end. So what we can say here is a greater percentage of patients died of heart-related problems with the biological valve in comparison to the mechanical valve, and that there is your final mark. One risk of mechanical valves is that blood clots can form on the surface of the valve. Name the component of the blood that starts the process of blood clotting. So the answer to this is platelets, and these are little fragments of cells in the bloodstream that can get sticky and form a kind of patch over a bleed. The mark scheme will also allow you to give the alternate name for them, which is thrombocytes. Evaluate the use of mechanical replacement heart valves and biological replacement heart valves. Use the information from Table 4 and your own knowledge, and this question is worth 6 marks. Now, with a question such as this, it's not enough to just list 6 bullet points to get all of those marks. You've got to make sure that you're listing your points in a logical order and explaining them for the examiner. The question then goes on to tell you to use information from Table 4 and use your own knowledge, so you must do both of these in order to get your full marks. And finally, you have to arrive at a judgment at the end of your argument, so you have to use the statements that you've just made supporting each type, and then you have to make a decision. So a way you might go about structuring this is making points for mechanical and then biological, or vice versa. So you can pick one and start to list some of its advantages and explain them, and then you can talk about some of the disadvantages. So for mechanical, you might say it's less likely to need a replacement within 6 years. That's 2.2% is less than 5.2%, which is a piece of data from the table if you look at the second to last row. You could also go on to say that they have fewer deaths from heart-related problems after their valve replacements, or lower percentage, because this is what we just calculated in the previous question. Next, you might start to go on to list some of the disadvantages. So you use a connective to turn your argument around. So we've said, however, blood clots in the brain are more likely to occur. And then you back that up with some data from the table. So we're looking at the last row and saying that 5.8% is greater than 0.1%. So that's a disadvantage. And this particular statement gives you an opportunity to insert some of your own knowledge. So if the patient is likely to have blood clots, they're going to have to take some anti-clotting medication, and this is likely going to be the case for the rest of their life. So that gets you your own knowledge points for that argument. If you want to take that even further, you can then talk about some of the side effects of the medication. So if they're taking anti-clotting medication, it can make their blood thinner, and that makes them more prone to excessive bleeding and hemorrhaging. In terms of the biological valve, there isn't any additional medication that's needed, so that is an advantage in comparison to the mechanical valve. However, a pretty big controversy with the biological valve is the fact that it comes from a living animal, it comes from a pig. And the pig doesn't get a choice in this, it can't consent, which gives some people some ethical or religious concerns. Another argument is that this biological material may be rejected by the body of the person receiving the valve, so it's more likely. And while we haven't got any additional medication in terms of the clotting, some patients may need to take immunosuppressants to avoid this problem of rejection. Lastly, now that you've made your points, you want to arrive at a final judgment, and this is based on your opinion, so there isn't a specific thing that you have to say in the mark scheme. As an example, we could say that we think the biological valve is better because it doesn't cause so much clotting, and the clotting can cause further problems that might be life-threatening, such as heart attacks. Finally, just to add the last little bit of strength to your argument, you can talk about the medication. So, because the patient doesn't need so many blood thinners or any anti-clotting medication, this means that heavy and potentially fatal bleeding is less likely, so particularly if they're in an accident or something like that, it's safer for the patient. People with diabetes have difficulty controlling their blood glucose concentration. Which part of the blood transports glucose? Tick one box. So the answer to this is plasma. This is your main transport medium and makes up quite a lot of your blood. The others all have different functions. So lymphocytes are part of your immune system, platelets are part of the clotting process, so they're little fragments of cells, and red blood cells are responsible for carrying oxygen. Glucose is often found in the urine of people with diabetes. Name a chemical used to test for glucose. And the answer to this is Benedict's solution. Describe a test that could be used to show that a person's urine contains glucose. So name the test and describe what the positive result is. So the test for this is adding Benedict's reagent to the sample, and then they can heat it up or boil it. And then the positive result is a colour change from blue to a brick red. The body cells of a person with untreated diabetes lose more water than the body cells of a person who does not have diabetes. Explain how diabetes can cause the body cells to lose more water, and this question is worth 3 marks. So this question is asking us about the movement of water, which means that a lot of your marks are likely going to come from stating osmosis and defining it. So first we're going to describe the conditions for this osmosis. We say the blood is more concentrated than the fluid inside the cells. Then we say that as a result of this, water moves from the cells into the blood by osmosis, and state that this is through a partially permeable membrane, so that would be the membrane surrounding each of the cells. So that's all you've got to say for your 3 marks. You talk about the concentration difference, which creates a concentration gradient. You'd state the term osmosis, and then you mention the partially permeable membrane, which is part of your osmosis definition. Glucose is absorbed into the blood in the small intestine by both diffusion and active transport. Describe how the small intestine is adapted for efficient absorption, and this question is worth 5 marks. So here you want to aim for 5 logically linked points. The first point you might decide to make is about the villi, and these line the surface of the intestine to provide a large surface area for diffusion. What they actually are is just these little projections, almost like fingers sticking out of the lining. The other important thing about these is that they have a very thin walls, very thin lining, and this makes a short diffusion distance, which also helps with the efficiency. Another thing that you could mention is that the small intestine is a very long tube, and this is important because it means that the stuff that is going to be digested has a long way to go, and so it's in there for a long time, and by giving it that bit more time, there's more opportunity for the absorption to occur. Then you could mention that there's a really good supply of blood in the intestines, and what this does is maintain a steep concentration gradient for diffusion to occur along. Lastly, you want to make sure that you're also mentioning the active transport, because the question does say both diffusion and active transport. So what you can say is that the cells have many mitochondria for aerobic respiration, and this provides them with the energy that they need to carry out active transport. And that will get you your 5th statement. A small animal called an axolotl lives in water. The axolotl has a double circulatory system. Define the term double circulatory system. So this is a system when the blood is pumped to the lungs by one side of the heart and pumped to the body by the other, so it almost works in 2 loops, hence double. And the advantage of this is that it keeps the oxygenated blood separate from the deoxygenated blood. Explain why having only one ventricle makes the circulatory system less efficient than having 2 ventricles. So when all of the blood is in one ventricle without a septum to separate oxygenated and deoxygenated, the 2 types of blood will mix together. And the problem with this is that there's a less efficient delivery of oxygen to the tissues of the body because it's getting back some of the deoxygenated blood that it's already used. Figure 8 shows an axolotl. Explain why an axolotl may die in water with a low concentration of oxygen, and this question is worth 4 marks. So when there's a decreased concentration of oxygen in the water, the concentration gradient of the oxygen becomes shallower. The reason for this shallower concentration gradient is the fact that the difference between the concentration of oxygen in the water and then the concentration of oxygen in the axolotl itself is smaller. And the overall effect of this is the rate of diffusion into the gills of the axolotl is going to be slower. Next, you want to go on to talk a little bit more about the effects of this. So less oxygen diffusing into the gills of the axolotl will result in less oxygen making it into the creature's bloodstream. So this oxygen is needed in the bloodstream in order for the axolotl to carry out aerobic respiration, so the type of respiration that does use oxygen. And when this can't happen, less energy is going to be released. And without that energy, less metabolism can take place. So metabolism describes all of those chemical processes that go on inside the body to keep it functioning normally and to keep the organs working. Now here you can see that each red tick is a relevant point, and so you need to have at least 4 relevant points to get full marks. However, there are some alternative points that you can make. So what you could say is that anaerobic respiration, that's the respiration that happens without oxygen, will be occurring and lactic acid will build up. And lactic acid is a kind of toxic byproduct, and so this won't help the axolotl survive either. If a gill of an axolotl is removed, a new gill will grow in its place. Scientists hope to use information on how axolotls grow new gills to help with regenerating human tissue. Name the type of cell that divides when a new gill grows. And the answer to this is stem cells. Name one condition that could be treated using regenerated human tissue. So this question has quite a few possible answers that you can give. You could say paralysis, so this is often the result of a stroke or a spinal cord injury, so that's the kind of cells that you'd be replacing. There's diabetes, so type 1 for example is caused when there's damage to the beta cells in the pancreas. Parkinson's disease is to do with the loss of nerve cells. There's heart disease where some of the heart cells or the arterial cells are damaged. And then you can also treat patients with cystic fibrosis, cancer, or even physical burns. Suggest one reason why an axolotl is a suitable animal for research in the laboratory. So an axolotl is very easy to breed. We could say that they're cheap to keep in that lab environment, mostly because they're so small and they take up very little space. So any of these points will get you your mark. An axolotl may not be a suitable animal to study when researching regeneration in human tissue. Suggest one reason why. So the first thing that might come to mind here is that the axolotl was a kind of amphibian creature, it's not a mammal like we are. We also got the information earlier that the part of the axolotl that is being regenerated is the gill, and we don't necessarily know that the gill regenerates in the same way to the rest of the organs, so that might not be possible even for the axolotl if it was the heart or something like that that needed regenerating. And the last argument that you might make is that the body processes and the metabolism in this little amphibian creature are just too different from those in a human. Pancreatic cancer develops when a malignant tumour grows inside the pancreas. The pancreas produces digestive enzymes. What is an enzyme? So the way that the mark scheme is looking for you to define this term is as a chemical which catalyses, and catalyse just means to speed up a reaction without being used up itself. So this enzyme catalyses the reactions happening in living organisms. And the simplest way that you can actually define this is by calling it a biological catalyst. Carbohydrase is an enzyme produced by the pancreas. Name 2 other organs in the digestive system that produce carbohydrase. So one of these organs is your salivary glands. So these are found in the mouth and they're particularly active when you're chewing. And the other organ that you can give is the small intestine. One symptom of pancreatic cancer is weight loss. Explain how pancreatic cancer may cause a person to lose weight. Do not refer to hormones in your answer. And this question is worth 4 marks. So let's just remember the significance of the pancreas. So here we're producing a lot of enzymes. Now, when you've got a patient with pancreatic cancer, they may be struggling with a reduced enzyme production. So they end up without a lot of their digestive enzymes, and the problem with this is that their food can't be properly digested to get the nutrients out of it. And what you can talk about particularly here is the glucose, so there's less glucose absorption into the bloodstream. This leads to less glucose being available for respiration, which is an energy producing reaction. However, the body still needs to get that energy from somewhere because it's got to carry on metabolizing, and so what it does instead is use up those reserves of body fat and hence the weight loss. Now we've actually made our 4 relevant points. Each red tick is a relevant point that can get you a mark. However, there are some alternatives that you can give here. So in the examples so far, we've talked about the effects of less glucose being absorbed. But you can also talk about the amino acids. So amino acids are the smaller subunits that make up proteins. You might state that fewer amino acids are being absorbed into the blood and fewer amino acids are being made into proteins for growth and repair. That's another reason a person might be losing weight. Similarly, you could be talking about the fatty acids, so this is another alternative second half of your answer. So less fatty acids are being absorbed into the bloodstream, just like we've said with the glucose and the amino acids, but this means that there's less fat that's now being stored in the body, there's less being uptaken, and now there's less being deposited as extra. And the last thing the mark scheme will allow you to mention is the chemotherapy. So this is the treatment that some cancer patients will be receiving, and it can cause them to have nausea and a loss of appetite. One thing to just be aware of here though is that this is a response to the treatment of the cancer, not so much directly a response to the cancer, so make sure you're aware of that if you make a note of it. Enzyme A and enzyme B are involved in controlling cell division in pancreatic cancer cells. Most cancer cells produce both enzyme A and enzyme B. Some people have a gene mutation that stops cancer cells from producing enzyme B. Figure 9 shows how cell division is controlled in pancreatic cancer cells. So here's figure 9 and it has a kind of flowchart structure. So on the left you've got the pancreatic cancer cells, and then when you're going upwards it's for the people with enzyme A or enzyme B, and the cells are dividing uncontrollably. In the scenario down and right, the cells stop dividing and are destroyed. Scientists have developed a drug that inhibits enzyme A. The drug is given to pancreatic cancer patients who have the gene mutation that stops cancer cells producing enzyme B. The drug only targets cancer cells. Explain why the drug can be used to treat pancreatic cancer patients with the gene mutation. Use information from figure 9, and this question is worth 3 marks. So in this person, enzyme A is being inhibited, and then they've already got the mutation that stops cancer cells producing enzyme B. So that means there's no enzymes allowing the cancer cells to divide. So that's your first mark, cancer cells cannot divide. As a result of this, the tumour doesn't form, or if they've got an existing tumour, it doesn't grow any larger. This drug can also help to prevent the spread of the cancer because it stops the patient from producing secondary tumours around the rest of the body. And for that last mark, we just have to clarify what we were thinking about earlier. The cells cannot divide because both enzymes A and enzymes B are not working. Explain why the drug could not be used to treat pancreatic cancer in a patient that produces both enzyme A and enzyme B. So this is because enzyme B would still be produced. This patient doesn't have the gene mutation that stops the cancer cells producing enzyme B, and this means that the cells will still divide uncontrollably to make that cancerous tumour. The drug was trialled before it was licensed for use. To improve validity of the results in the trial, some patients were given a placebo and a double-blind trial was used. Give reasons why a placebo and a double-blind trial were used. So if we first define this term validity, so this is how accurately the experiment measures what it was intended to measure. A placebo is a dummy drug. It's given to the patients that don't get the actual drug, and it's partially there for the comparison, but it also takes into account the psychology of the patients. Because there is something called the placebo effect, which is when the patient gets something and it doesn't necessarily have to be having any real effect on their symptoms, but they believe it does. A double-blind trial is when neither the doctors nor the patients know who has received the placebo and who has received the actual drug, and this is done to avoid bias. One stage in a drug trial is to test the drug on healthy volunteers. What is the next stage in the drug trial? Tick one box. So they've just tested on healthy volunteers, most likely looking for side effects. The next thing they've got to do is actually see if the drug treats the disease in the way they expect it to. But they can't just start rolling the drug out to everybody because they haven't tested that last bit yet. So they have to wait for some volunteers with the disease, which is that last box there. A monoclonal antibody has been produced to treat pancreatic cancer. Explain how the monoclonal antibody works to treat pancreatic cancer. And this question is worth 3 marks. So let's recap. Monoclonal antibodies are identical copies of one type of antibody. So how do these work when treating the pancreatic cancer? Well, what you need to say here is that the monoclonal antibody is attached to a radioactive substance, and by radioactive substance we mean whatever's actually attacking the cancer. So the mark scheme will allow you to put toxin or chemical or drug. It's just what's being used to kill those cancerous cells. Now, monoclonal antibodies are specific, and so for your next mark, you've got to say that the monoclonal antibody will only attach to the target, and for this you can say cancerous cells or you can call it the tumour. The mark scheme doesn't mind either of them. Lastly, you say that the radioactive substance or toxin, drug, chemical, if you said that, the radioactive substance will bind to the cancer cells and it will stop them from dividing. There are a couple of alternatives to the last marks, and these are for describing how the monoclonal antibodies stop the cancer cells from continuing to divide. So you can say that the monoclonal antibody interrupts the cell cycle, or you could say that the monoclonal antibodies help the immune system of the patient to destroy the cancer cells for itself. But overall, each bullet point is a relevant point, so as long as you've got 3 of them and they're sequenced logically, you can get those 3 marks.